All right, so this is the last lecture for general biology. This one is gene expression. So you have a cell here, right? Let's draw a cell. Let's draw a cell with a marker that actually works. Here we go. And in your cell you have most of your DNA kept within the nucleus of your cell. DNA provides the blueprint by which proteins are built, and those proteins make up who you are. So the proteins are actually going to be built using ribosomes out here in the cytoplasm of the cell. The DNA never leaves the nucleus of the cell, which means we need a middleman in this process. And the middleman is called messenger RNA. It's a single-stranded molecule, a single-stranded nucleic acid that is going to be a copy of a particular segment of DNA called a gene. If you remember from before, genes are segments of DNA that code for a particular trait. So in this case, messenger RNA, it finds a particular region of DNA that codes for the gene that we need to build a protein based on. And it takes that gene, copies the information here, and brings it out. Think of it like DNA is a very, very long book, and messenger RNA is taking a single chapter out of it to carry out to the cytoplasm of the cell. So, DNA to messenger RNA, that process is called transcription. And if you think about it, transcribing something simply means copying, but usually it refers to copying in the same language. So if you miss a day of class and copy down a classmate's notes from that day, you are transcribing them. It's probably still going to be in English. However, going from messenger RNA to a protein, that's going from the language of nucleic acids to amino acids, which is what makes up proteins. That's very different. And so that's going to be translation. It's like you're going from one language to another. You're translating the messenger RNA into the language of proteins. Transcription and translation together form what's called the central dogma of biology. This is how the genetic information of living things determines physically what or who they are, right? Blueprint, photocopy of the blueprint, used to build the organism in question, because living things principally are built from proteins. So transcription, we're not going to look too much at, but with transcription, you have a section or a segment of DNA, right? And DNA is found on chromosomes, which have hundreds upon hundreds of different, even thousands of different genes. And so the RNA has to find the particular gene that we're interested in. It finds a region of that DNA called the promoter region. That's the start of the gene. It synthesizes what we need starting at that promoter. Let me draw that for you real quick. So here is a small segment of DNA, double-stranded, right? Here is the start of a gene, here is the end of a gene. The start of the gene is called the promoter. The end of the gene is called the terminator region. What happens is RNA polymerase is going to ultimately separate the two strands of DNA, starting at the promoter and ultimately ending at the terminator region. And it's going to produce a single-stranded copy of the information from the DNA. And if you remember how DNA works, it has the nitrogenous bases A, T, C, and G. And so the messenger RNA, using the rules of specific base pairing, only needs to see that one letter and say, oh, these are the complements that I need. C always goes with G. A always goes with T. So messenger RNA creates this little segment that corresponds to a particular gene, and then it leaves the DNA zips back into place, and we have our photocopy, so to speak, of genetic information in the form of messenger RNA. Now, the first thing that has to happen after this is that we have to remove what's called junk DNA, also known as introns. Basically, junk DNA or introns are segments of DNA that don't actually code for the protein. If you want to think of how that might work, let me give you something here. Take a look at those two unintelligible things. So many useless letters. We need to get rid of the introns. There are... Oh, I accidentally deleted one I should not have deleted. That was there. There was a C there. So, now we have 6 letters. Splice them together. And you get these 2 words, the cat. So many useless letters in between the letters that actually mean something. That is the idea of junk DNA. It's a bunch of letters in your DNA that aren't necessary to be read by the ribosomes and translated into a protein. They estimate 98% of your DNA is actually junk DNA, which I think is fascinating. But once you get rid of the introns and splice together the actual coding letters of your DNA, then we are ready to translate that DNA. I've got a video here that I think is going to be the best way for us to look at this. So let's kill the lights and go through the process of translation. The virtual cell presents translation. Translation is the synthesis or production of a protein using a messenger RNA template. And we're going to use several key molecules in this process. This first one is messenger RNA. Now, there's a methylated cap, which I don't care that you know about, right here. But I want you just to focus on this here. This is the strand that we drew, which you probably can't see right now. But it's the strand that we drew that came from a small section of DNA for a particular gene. So this is a strand of RNA. This is a ribosome. It has a large and small subunit. This is the protein factory. This is where the actual translation of the messenger RNA will occur and where the protein will get built. This is transfer RNA. It has an important role to play, and I want you to just keep it in mind as we go along. tRNA, transfer RNA. We're not worried about the release factor. We're also not worried about the stages of this. So this is messenger RNA. Now I want you to look at what it's doing here. It is separating messenger RNA into 3-letter combos called codons. A codon is a snippet of 3 bases in messenger RNA. So what are some examples? Well, one example would be ACG, or UUG, or CAA. There are a whole host of them. This next one could be GGC. All of those are codons, 3 letters together. There are quite a few possibilities out of the 4 A, C, U, and G that we have in RNA. So we've got letters being read as clusters of 4 in the messenger RNA. Notice how many codons there are. There's quite a few. So the messenger RNA is now entering the ribosome. One side of it is going to be read first. It's going to be sliding through the ribosome like this, with each codon being read as it goes through. The first codon is going to be AUG. I can tell you that one right now. All right, and so it's going to read that codon and respond accordingly. How's it going to respond? It's going to respond by bringing in the appropriate amino acid. I need to show you a chart. This is a handy-dandy codon translation chart. Messenger RNA is translated into proteins. Messenger RNA is read as codons, 3-letter combinations. Each letter combination translates into a particular amino acid. And if you remember anything from the macromolecules chapter, proteins are built from amino acids. Different proteins have different combos of the 20 types of amino acids. And so, determining which protein gets built comes from the arrangement of the codons in the messenger RNA, which ultimately came from the letters in your DNA. So look at this chart. AUG, I told you, is going to be the first codon. AUG, methionine. It's the start of every single gene. Right? So let's go ahead and go back to our video. The codons are going to go into the ribosome and this molecule, if you remember it, this was transfer RNA. Transfer RNA is going to carry in the appropriate translated amino acid for whatever codon comes through in the messenger RNA. Let's go ahead and work through this. Don't pay attention to a lot of the words that they're going to flash. I just want you to watch what happens. Transfer RNA comes in. it has an anticodon. I'm not too worried that you know that. But notice that we have AUG here. AUG is the first codon. AUG is translated into methionine, the amino acid methionine. And so the transfer RNA brings in the methionine amino acid, which it represents here as just what looks like a myconite. Sounds incredibly good right now, by the way. This is the other part of the ribosome here. There are actually two sites where transfer RNA can come in, so it can read effectively two codons at once. The second codon in this particular messenger RNA chain is UUU, which is going to translate to a certain amino acid that transfer RNA has brought in. The transfer RNAs will do their job and then move on, but they leave behind their amino acid and it starts to form a long chain, a polymer, building up the protein. So let's watch the messenger RNA slide through the ribosome and watch how each codon is read and the appropriate translation is brought in. So the transfer RNAs leave, they leave behind their amino acid. Each codon is read and the appropriate amino acid is brought in that corresponds to the 3 letters of the codon. And this happens, and we build a chain of amino acids, which is a polymer. Each amino acid is called a monomer. And that happens all the way until we get to the end of the messenger RNA strand, where there's going to be a codon that does not code for any particular amino acid. It's called a stop codon. It calls for the release factor to signal this is the end of the messenger RNA strand. Our protein is complete. The protein has been built. The ribosome breaks apart. And we have our protein that has been built from the instructions given in the messenger RNA strand. So that is translation. Right? And you're built from proteins. This is how your DNA determines what you look like. Sections of your DNA have genes. Those genes are transcribed into messenger RNA, which is then translated into the proteins that build you. Sometimes there are mistakes that happen, mutations. If the mutation happens during the formation of an egg cell or a sperm cell, it's called a germ cell mutation. That's how you get mutations that people are born with. Most people are born with some mutations. Usually they don't have any effect. Some are harmful, some are beneficial, and natural selection favors them. But how a mutation works is that one of the letters gets changed. One of these letters gets changed. So let's say that we were supposed to have UCG, that's our codon. UCG. This is the codon that was supposed to be present in a particular section of messenger RNA. That codes for, or is translated into, the amino acid serine here. But let's say there's a mutation in the copying process, and instead of having UCG, we end up with UCC. UCC still codes for the amino acid serine. Even though the actual letters have changed, it doesn't change which amino acid is coded for. This is called a silent mutation. Silent mutation. What if there is a mutation that goes from UCG to CCG? Now, that's translated as proline. We have changed the amino acid. All the other amino acids in that particular protein are going to be the same, but this one is different. That's called a missense mutation. This mutation, you wouldn't think it would have a lot of effect, but it can. Sickle cell trait, which causes the red blood cells to have that sickle-like shape, that's caused by an ancestral mutation whereby one letter was changed in the gene that ultimately determined the shape of the blood cell. The problem was that letter changed that one amino acid from one that was not hydrophobic to a hydrophobic amino acid, which means the red blood cell is trying to physically avoid water. Well, blood plasma is mostly water, and so it creates that sickle shape as the one different amino acid is responding to the blood plasma very differently than it should. Again, sickle cell trait persists because it offers defense against malaria, but ancestrally, that's how the mutation got there in the first place. Somebody had a single letter change, a missense mutation that occurred. What if UCG had a mutation that caused UAG? This would result in the stop codon, which means everything that was supposed to happen after this serine does not actually become, it's not made. And so we have premature end of translation. The protein's not as long as it should be, right? This is called a nonsense mutation. All three of those mutations are examples of base substitution. Base meaning letter. The nitrogenous bases are the letters. So we're just swapping one letter for the other. There's another kind of mutation called a frame shift mutation that is often a disastrous mutation when it occurs. I'm going to give you a sentence, all with three-letter words. The cat ate the rat. A base substitution mutation might do something like this. The cat are the rat. Changes the meaning of the sentence. It's not really sensible anymore, but only one letter was altered. It can still affect the other things around it, obviously, but it didn't actually change the other amino acids translated from these codons. A frame shift mutation involves adding or deleting an extra letter which shifts the frame of the entire codon reading. So let's say we inserted the letter M right here. Okay, we've replaced one letter, but what about the rest of this? Well, the E just got pushed over. So now, everything is off. You can notice how, if you were to put these little imaginary lines here, you can make sense of what it's trying to say. But it's not read like that. It's still read as three-letter codons. And so we've just thrown off the entire frame. We've shifted the frame. And now it's no longer really intelligible. That's what a frame shift mutation is. Usually they are not selected for. Usually they are disastrous because they change so much about the actual gene that's in question. That is a summary of gene expression. And this is the last lecture that you guys will be listening to. I hope you guys have a I don't know if I can switch this thing here. Let's see. Yeah, I'm just going to turn this off before I screw up the recording process. Bye, everybody.